Hello, biology. The purpose of this video is to give students an opportunity to finish the notes if I went too fast in class, or if students were absent, to give them an opportunity to take the notes. If you are starting from scratch, you are going to need a Google Doc with 16 rows and 2 columns. If you're doing it by hand, of course, draw a line down the center of your page, and please do not forget to number your notes. I'm not going to spend as much time as I do in class on these notes. I will point at the camera when I'm getting ready to advance to the next slide. So obviously, if you are still writing or typing at that time, that would be an opportunity for you to pause the video. All right, here we go. Genetics, fun stuff. All right, so the genetics, it is a sub-discipline of biology. It is the study of heredity and how characteristics are transferred from the parents to the offspring. Hi, Gregor Mendel was a monk. Apparently he had a lot of time on his hands. His work helped with the understanding of heredity and how traits are transferred again from parents to offspring. His work, he used garden peas. He used garden peas for a couple of reasons. Number one, they have some very easily identifiable traits. They also don't take up a whole lot of space, so you can do literally hundreds or thousands of crosses. And they reproduce pretty quickly. So they actually, what's called go to seed, meaning the plants mature, and after fertilization, they create seeds. They go to seed pretty quickly. So those are all characteristics when you want to do a whole bunch of trials. And again, we talked a lot about fertilization when we were going over meiosis. So you have an egg cell from the female parent, you have a sperm cell from the male parent, and once the sperm cell penetrates the egg, it takes the father's chromosomes, combines them with the mother's chromosomes, and it forms a new cell. Okay, that cell we call a zygote. And remember, after fertilization takes place, the two haploid daughter cells, the sperm and the egg, now form a diploid zygote cell. And then what happens is it goes through rapid mitosis, and it creates something that's called, I think, if I remember correctly, a blastocyst. And then cells start to specialize, and then you eventually get an embryo, and then the embryo goes into a fetus, and then the fetus grows into a baby. All right, moving ahead. Okay, so self-pollination. Many plant species have both the male and the female reproductive structures on the same plant. Okay, obviously humans and many animals, that is most animals, that's not the case. So self-pollinating is when the male pollen, okay, that's the sperm cell, a plant sperm cell. So for those of you that have allergies to pollen, you're allergic to male sperm, basically male plant sperm. What happens is the pollen on one plant fertilizes the female structure on the same plant. And of course, you're going to get offspring that are identical to the parent plant. All right. True breeding, same as pure breeding, means that you will get a clone identical to the parent, okay, if self-pollination is taking place. You still are producing egg cells and sperm cells through meiosis. You're just simply not, it decreases genetic variation when you are self-fertilizing or self-pollinating. So then what Mendel did is he took pollen, he used a paintbrush, took pollen from one plant and used it to fertilize the female structure on another plant. And of course, whenever you have that, just as with animals and humans, if you have a child or have offspring and the two parents are different, you can expect characteristics different from both parents. Obviously, the offspring is going to have some things in common with each parent, but you will not get a clone of the parent because you're taking chromosomes from one organism, genes from one organism, and combining them with chromosomes and genes from another organism, and you get a hybrid, a combination of traits from both parents. Moving ahead. Okay, so what happened? It produced seeds from two different plants. And again, we call these hybrids. Many of you are familiar with the term hybrid in terms of a car. It's a hybrid because basically it's got characteristics of two different types of cars. It's got characteristics of gas-powered car, and it's got characteristics of an electric-powered car. My bicycle is a hybrid. It is a mountain bike, so it's got the stronger frame. It's got the shocks in the front. It's got the funky, mountain bike handlebars. But then I put street tires on it so I could go faster when I'm riding on the street and on the bike trail. So it's got characteristics of two different types of bikes. And again, if you are taking notes on this slide, I would recommend doing a screenshot or a snippet. The pictures on these notes, it makes the notes create more sense for the reader, for the note taker. If you have something to look at that kind of shows you what's going on. So this shows the paintbrush taking the pollen from one plant and fertilizing the female structure on a different plant. All right, moving ahead. All right, some vocabulary. Trait, which is basically synonymous or the same thing as a characteristic. It varies from one individual to another. Excuse me. Okay, I'm going to be using these terms a lot for the next 4, 5, 6 weeks. The P generation is the parental generation. And of course, the parents will be a pair of plants. Or if we're talking about humans or animals or the parents, two different parents. The F1 is the first filial generation. So the first set of offspring is the F1 generation. And then hybrids, these are offspring that come from crosses between parents with different traits. So the offspring that were created from the seeds when he did his cross-pollinating, taking pollen from one, fertilizing the female structure on another, you are combining the characteristics of those two different parents, and you can expect the offspring to have a combination of traits in common with the parents, but they will not be identical to one parent or the other parent. So we'll get into this in just a second, but when he took the first trait he was looking at was tall versus short. When he took a tall parent, okay, and he took a short parent, all right. So again, we're talking about a hybrid, okay. All of the offspring in the F1 generation, the first generation, were tall, okay. And I'll kind of give you a little hint as to what we're going to talk about. That's because the tall parents gave the dominant allele, capital T in this case, and the short parent gave the recessive allele, little t, and all of the F1 generation were heterozygous, big T, little t. And of course, whenever you have a big T or capital anything, you're going to see the dominant trait. So this probably left Mendel scratching his head a little bit. It's like, I got a tall parent, I got a short parent, but all the offspring are tall. What's going on? And it's because he didn't understand the different alleles. He didn't understand dominance versus recessiveness yet. Any of it. Moving ahead. Okay, so these were the traits he was studying. Round, dominant, yellow, dominant, gray, smooth, all dominant. All right. Wrinkled, green, white, constricted, all recessive. Okay, so these were the crosses. All right, dominant parents crossed with recessive parents, and in each case, he got the dominant trait. So what happened to the alleles, or what happened to the genes that the recessive parents contributed? Moving ahead. All right, here is 4 different characteristics that he was studying. So pod shape, pod color, flower position, plant height. Again, dominant traits being crossed with the recessive traits. And in every single case, all of the F1 offspring had the dominant trait. All right, so what's going on with the recessive trait? They were being dominated. These are all heterozygous F1 hybrids. And of course, heterozygous means you have both the dominant allele and the recessive allele together. And whenever you have the dominant allele, you get the dominant trait. Moving ahead. All right, please use this as an opportunity to practice a little bit with your notes, see how much you've learned already. Let's do a little bit of matching. And of course, this goes back to number 9, 5, 6. All right, here we go. Fertilization. D, sperm and egg cells join. Cross-pollination. A, pollen used from a different plant. Trait. E, characteristic that varies. P1. B, first set of parents. F1. How appropriate. F, first set of offspring. Hybrid. C, their parents have different characteristics. And remember, all the hybrids in his cross-pollination experiments had the characteristic of the dominant parent. That's because they only got a dominant allele from the dominant parent, so the dominant parents were homozygous dominant. And whenever you get a dominant allele, regardless of what the second one is, you're going to get the dominant trait. When true breeding parents were used, what parent did the offspring look like? When true breeding parents were used, what parent did the offspring look like? And again, this is self-pollination. They look like the parent. Because remember, true breeding, self-pollination, there's only really one parent. There's a parent with a male structure on it, and the same parent has the female structure. So it's basically cloning that parent. Moving ahead. Mendel's first conclusions. That inheritance is determined by factors. And of course, these factors are genes, and these factors we refer to as alleles. There are 2 different factors. Well, for the most part, there are 2 different factors, a dominant or recessive. And the genes get passed from one generation to the next. Okay, so the idea about alleles, or different forms of these genes. Each of the traits he studied was controlled by one gene that occurred in 2 different forms. And those 2 different forms, of course, were dominant form and a recessive form. Alleles are different forms of a single gene. All right, I'm moving ahead. All right, a second conclusion, which is the principle of dominance. And you learned about what the principle of dominance was in today's warm-up. I believe it was warm-up question number 35. Some alleles are dominant. We're going to abbreviate that as DOM. And others are recessive. We're going to abbreviate that as REC. And the recessive ones are dominated. All right, so the difference between the two. If the dominant allele is there, the dominant trait will always show. Simple enough. If the recessive allele is there, the recessive trait will only show if it is there twice, or if it is there without the dominant allele. And that was what was in warm-up question number 36 today. All right, moving ahead. Okay, segregation. All right, so segregation, obviously, in terms of the civil rights movement and slavery, etc., was talking about separation. Okay, colored people, African Americans, were not allowed to sit with, use even the same drinking fountains, use the same bathroom as white folk. All right. In terms of genetics, segregation is referring to gamete formation. When sperm and egg are formed, the two alleles, because remember, everybody is diploid, every parent cell is diploid, but after meiosis, those chromosomes, which we can kind of equate to alleles in this case, get separated. And each sperm cell has only one allele, and each egg cell has only one allele. And that's really important because that is the only way for a recessive trait to be shown is if you have one allele. So let's call this the egg cell, which is recessive. And then this other allele, which we'll call the sperm cell, is recessive. The only way you can get a recessive trait is if both of those alleles in each individual parent get separated from a dominant allele, and then during fertilization, they can come together to give you the recessive trait. So we crossed the F1 generation. So remember, the F1 generation, those were all hybrids. They were all heterozygous. So he self-pollinated the F1 generation, and I have no idea what logic led him to that experiment. Maybe he was just stupid smart. The F1 generation with a self to produce an F2, the second filial generation, so the second generation of offspring. And the traits controlled by recessive alleles reappeared in 1/4 of the plants. So let's just say we had 1,000 offspring in the F2 generation. That means of the 1,000 offspring, approximately 250 of them were short. And of course, that means 750 of them were tall. All right. So again, probably left scratching his head saying, okay, tall versus short, F1 generation, they're all tall. All right. Self-pollinated these tall ones, which is exactly what he did with the tall and short ones individually. And he got all tall when he self-pollinated the tall, and he got all short when he self-pollinated the short. But he self-pollinates this F1 generation, and 3/4 of them are tall, and 1/4 of them are short. So this led him to believe that during gamete formation, sperm and egg formation, for those F1 hybrids, the dominant allele got separated from the recessive allele, okay, which is segregation. And then that gave an opportunity for a recessive sperm and a recessive egg to get combined and produce a seed that would grow to a short plant. My bad. Moving ahead. Okay, so this is what it looked like. Tall, short, okay. In the parental generation, the F1 generation, they were all tall. All right, so what he did is he self-pollinated, so it was a tall times a tall. And check that out. 3 tall plants, 1 short plant. This led him to believe that when gametes were being formed, okay, sperm and egg, okay, during the self-pollination, the dominant trait or the dominant allele got separated from the recessive allele. The only way for you to get a short plant is if you get a recessive allele from the male and a recessive allele from the female. Moving ahead. His thoughts about the recessive allele in the F1 generation. He assumed that a dominant allele had hidden or dominated the recessive allele in the F1 generation. That's why all the plants were tall. Because remember, they were all heterozygous. Big T and a little t. All right, a big T and a little t. Whenever you got the big T there, you are going to see the dominant trait. The trait controlled by the recessive allele showed up in some of the F2 plants, and of course now we know that's because when the sperm cells were created, half of the sperm had the recessive allele, half of the sperm had the dominant allele. For eggs, half of the sperm had the dominant allele, half of the eggs had the dominant allele, half of the eggs had the recessive allele. Obviously, to get those short plants, you had to have a recessive allele egg, a recessive allele sperm, created the seed, grew into a short plant. Moving ahead. All right, so what happened? Let's summarize what happened. The return of this small trait controlled by the recessive allele indicated that at some point the allele for shortness had been separated or segregated from the allele for tallness. That happened during gamete formation in the F1 generation during self-pollination. That separation happened during gamete formation, and gametes only carry 1 allele for each gene, just like they carry 1 chromosome of the pair. So when we are talking about alleles, think of an allele representing a chromosome. Okay, half the chromosomes, half the alleles. All right, moving ahead. All right, so this is what it looks like in a diagram. So these are the F1 generation. They were hybrids. They were all tall, and they were all tall because we have this capital T. All right. Then when you self-pollinated, which basically means you're multiplying it by itself, during step 3 of a genetics problem, we separate the alleles. So this is going to be, we'll call this the male. Sperm cell with a capital T, sperm cell with a lowercase t. Call this the female. Egg cell with a capital T, egg cell with a lowercase t. And the F2 generation, 1/4 of them were short because they got a little t combined with a little t. Right? And I could represent this as a Punnett square as well. This is just a different diagram. Okay, so notice 3/4 are tall, 1/4 is short, but, okay, big T, you're going to have the same phenotype as big T, little t. They're both going to be tall. So what this means is there's really only 2 possible phenotypes, tall or short. But there are 3 possible genotypes. So if this was a genetics problem and I was asking for like what's the probability in a percentage of getting tall plants, that would be 75%. But if I asked what is the probability in a percentage of getting the genotype big T, little t, that would be 50%. So notice that more than one genotype can end up with the same phenotype, and that's simply because of the principle of dominance. As long as you have one capital T, you're going to have the phenotype of tall. Moving ahead. Okay, a blank allele for a trait is the one that is always seen, while a blank allele for a trait is the one that is hidden when grouped with which allele for a trait. So you're going to want to take a look back at number... Take a look back at number 12 and 13 for this one. Yeah, so it's a dominant allele for a trait is the one that is always seen, where a recessive allele for a trait is the one that is hidden or dominated when grouped with a dominant allele for a trait. What fraction of the plants in the F2 generation had the recessive trait reappear? Take a look at the number 14. Yeah, so 1/4 of the plants in the F2 were short, which is the recessive trait. You could also look at that as 25%, okay, of the offspring were short. Why did Mendel think that the recessive trait disappeared in the F1 generation? Take a look back at number 15. Yeah, because the recessive allele was grouped with a dominant allele. Therefore, there was no opportunity for the recessive trait to be seen. The recessive trait was dominated by the the recessive allele was dominated by the dominant allele. Okay, gametes are known as... You got it. B, sex cells. The offspring of crosses between parents with different traits, that F1 generation, what do we call those? Organisms, characteristics, two different parents. And technically, the F1 generation didn't have characteristics of the short parent, but it had the allele from the short parent. Those are called hybrids. In Mendel's pea experiments, the male gametes are, what do we call the male gametes in peas? And in most plants, honestly. Pollen, good. Okay, in a cross of true breeding tall pea plant with a true breeding short pea plant, the F1 generation, are they all short, all tall? Is it half and half? Or are they all medium height? Come on, we haven't talked about anything about being medium height. It was either tall or short. Which one was it? Yeah, the F1 generation, they were all tall. And that left him, probably scratches his head saying, wait a minute, that's a short parent. That's a tall parent. And all the offspring are tall? What the heck is going on? And of course, we now know that it's because the big T came from the tall plant and the little t came from the short plant. They're together and the little t got dominated by the big T. If a particular form of a trait is always present when the allele controlling it is present, then the allele must be, and this is a tough one, if it's always present, what does that mean about that allele? Means it's dominant. All right, ladies and gentlemen, thanks for being with me, and I will see you back in the Zoom.